In the previous video of glucose metabolism, we discussed about the glycolysis pathway. If you want to watch that video first, you can click the link above that slides in. In the glycolysis, we saw there was a breakdown of glucose molecule to the two molecules of pyruvic acid or pyruvate. Now this pyruvate molecule has two ways to go. If there is anaerobic condition, that's in the absence of oxygen in the cell, the pyruvate will go through fermentation process, which we have already discussed. The link is in the description. Or if there is aerobic condition, that's in the presence of oxygen, the pyruvate will go through a cellular respiration process, which goes through a cycle of reactions called the citric acid cycle and will eventually enter into the oxidative phosphorylation. So here today in this video we'll be discussing about the citric acid cycle, or you can say Krebs cycle. To start with the citric acid cycle, the pyruvate molecule formed from glycolysis in cytosol is first transported into mitochondria, as all the reactions of citric acid cycle, or you can say Krebs cycle, will be driven in the mitochondria. Here in the mitochondria, this pyruvate molecule is oxidized by oxygen molecule to carbon dioxide in a series of oxidation reactions collectively termed as cellular respiration. So what we get from this citric acid cycle? From this citric acid cycle, we will get high energy electrons. And these high energy electrons will be carried in two types of molecules called electron carriers in the form of NADH molecule and FADH2. So these two types of molecules, NADH and FADH, will carry out the electrons into the electron transport chain. Now let's start with the reactions first. From this glycolysis, we get two molecules of pyruvate. That means we have to start with pyruvate molecule. And it must be noted that citric acid cycle runs two times in order to oxidize the two molecules of pyruvate in each cycle. Then after we see the pyruvate formed in the glycolysis is transported from cytosol across the mitochondrial membrane to the matrix. And after that, pyruvate in presence of PDH enzyme, that's pyruvate dehydrogenase enzyme, reacts with coenzyme A forming carbon dioxide and the intermediate acetyl coenzyme A. And also during the pyruvate dehydrogenase reaction, NAD is reduced forming NADH molecule. Now from here the cycle begins. That means all the reactions from now are included in the citric acid cycle, with the first reaction coming as condensation reaction. Here first of all, 2 carbon acetyl group from acetyl coenzyme reacts with the 4 carbon molecule oxaloacetate in presence of citrate synthase enzyme to yield us the 6 carbon compound in the form of citric acid or citrate. The citric acid is the first compound formed in the cycle, after which this cycle gets its name. That's why it's called citric acid cycle, because the first carbon compound formed in this cycle is citric acid. Then we see the citrate is acted upon by aconitase enzyme and gets converted into cis-aconitate. And the next reaction is also catalyzed by the same aconitase enzyme, which further converts this cis-aconitate into isocitrate, which is a 6 carbon compound. Now after that we see this isocitrate is acted upon by isocitrate dehydrogenase enzyme, which converts it into alpha ketoglutarate. And in this reaction the NAD is also reduced to NADH molecule, and also 1 molecule of carbon dioxide is released in this reaction. That means this alpha ketoglutarate is only 5 carbon compound, since 1 carbon has lost in this reaction. Furthermore, alpha ketoglutarate dehydrogenase acts upon alpha ketoglutarate and there is a reduction of NAD into NADH molecule. And also here the 1 molecule of carbon dioxide is released. And we get a 4 carbon compound in the form of succinyl coenzyme A. Now this succinyl-CoA is catalyzed by succinyl-CoA synthetase and converts it into succinate. In this reaction there is addition of GDP plus inorganic phosphate, which yields us GTP, and also coenzyme A is lost from the succinyl-CoA. After that, succinate dehydrogenase catalyzes the succinate, and in this reaction FAD is reduced to the molecule of FADH2. And also from this reaction we get fumarate compound, which is a 4 carbon compound. And furthermore, the fumarate is acted upon by fumarase enzyme and converts it into malate, which is further catalyzed by malate dehydrogenase into oxaloacetate, a 4 carbon compound, from which we have started the citric acid cycle. And also in the last reaction of Krebs cycle, another NAD is reduced to NADH molecule. So this is how the cycle runs, all the reactions included in the citric acid cycle. Now let's have a look at the electron carriers reduced in the citric acid cycle. We see the first NADH reduced is in this reaction. Then second one is here, third one and fourth one. And we get only one FADH2. That means we get 4 NADH and 1 FADH2 molecule from one citric acid cycle. And we know this cycle runs 2 times. That means we will get 8 NADH molecule and 2 FADH2 molecule. And also it must be noted that we also get 2 GTPs from the citric acid cycle when it runs 2 times, since we get 1 GTP molecule from one cycle. So this is how much electron carriers we get from citric acid cycle, and these electron carriers will deliver these high energy electrons into the electron transport chain for the generation of ATPs through chemiosmosis. So this is all about citric acid cycle, or you can say Krebs cycle. I hope you like the video. If you like it, give it a thumbs up and make sure to subscribe this channel. Thanks.